Hey gang, this is a walkthrough of the DNA barcoding, DNA extraction part of the DNA barcoding. So once you open the procedure, you'll scroll down to part 2, and what you see at the beginning are the list of materials. Then you'll see two sets of instructions. One is a visual representation of the steps, and below them are the actual steps written out in text. So if I go up here, previously, you should have added your specimen to the tube and labeled the tube. It turns out that you're going to also need two other labeled tubes per specimen. So if your specimen is A1, then you have a tube labeled with A1 that has your specimen in it, but you're going to need two empty tubes that are also labeled A1 because you're going to transfer the DNA twice. So in the next step, you're adding the lysis solution, 300 microliters of it, to your sample. The lysis solution is going to break apart your cell nucleus, the cell membrane, and if it sits too long, it may also start to degrade your DNA. So once we put in the lysis solution, we need to go through most of these steps until the next stopping point. Because what we need to do is, you see here and here, we're going to add wash buffer, which is going to wash the lysis solution off of our DNA after it's had time to work. So I'm going to show some of these steps, talk through some of these steps. So here we have our sample with the lysis solution. You see that we're grinding. We need to grind it with this pestle, and we're going to try to squeeze it. Then we're going to put our sample in 65 degrees for 10 minutes. Then we're going to centrifuge it. This is a centrifuge. It's going to spin the sample. And what's going to happen is going to collect all the heavy materials at the bottom, and which is called a pellet. And then the solution on top is called a supernatant, which is the lighter things. Now notice it has to be balanced. So when you put one sample in one of the holes, you have to put another sample on the opposite end. Then you close the lid, you can cut it on, and it'll spin. So make sure you follow the directions for the amount of time. Once it's over, you just click the switch off and wait till it stops spinning. And you open the lid again. Now you see the pellet on the bottom. That's the heavy materials. And on the top is the supernatant, which is the liquid. So in some of these steps, you can see that we actually need to collect some of the supernatant. And we're trying not to touch the pellet at all. You can see we're transferring to a brand new tube. And this should have our DNA. We're adding silica, which is going to bind to DNA. It's a white solution, a white liquid. So we're going to pipette the silica. We're going to put it into our sample. And once we get that in, we're going to mix it around, which is vortexing. So we're going to try to break it around, shake it, pipette in and out to help mix, get air bubbles in and out, and just trying to mix, trying to get the white spread throughout the entire solution. So you see I'm pipetting there. At this point, we have already centrifuged this, so I'm not going to show that again, but you see the pellet at the bottom and the supernatant on the top. In this step, is interesting, is we're actually getting rid of the supernatant, so we don't want to disturb the pellet, so we're pipetting out. So you have to figure out how much solution is in there based on what you've added. You can squirt it out, and once you finish collecting, you get rid of the tip. And you see there's only the pellet. We want to keep that pellet, so make sure all the liquid is away from that pellet. See here, I'm skipping a few steps. We're adding wash buffer. And now we're also breaking up that pellet, vortexing, mixing it around to get that pellet broken and dissolved into that liquid. So you see there now, you can see it broken up, no more pellet. This is the wash buffer that's getting rid of that lysis solution. So we centrifuged it again, and we have the pellet in there again. So now we want to get rid of the supernatant to get you see we're getting rid of the supernatant. We're trying not to disturb the pellet. So you have to try to figure out how much is left. Here we're adding wash buffer to that pellet, and again, we're going to have to break it apart, pipette in and out. We're breaking it up, pipetting into breaking up that pellet. And then, we're going to repeat the step again. We've already centrifuged. Near the final step, we are actually taking some of the supernatant to not disturb the pellet, and we're going to transfer that supernatant to our final clean tube, and that will be our DNA that we're going to store for the next step. Now I want to show you just 3 major things I want you to think about throughout this process. The first thing is, if you look at this step, the initial incubation step where we're putting it in the 65 degrees, it has to sit for 10 minutes. Now, if you are a class that has the lunch period, this is a good time to eat your lunch during that double lab. If the solution needs to stay in for 12 minutes because you haven't finished eating, then I think that's okay. For the other sections, this is a good time to review or preview the upcoming steps. And in general, if you find yourself waiting, for example, when there's centrifugation or the solution's sitting in heat, there are things you could do to get ready for the next steps. For example, during this 10-minute period, someone could set a pipette or find the pipette that's going to give you 150 microliters, since you know that you're going to need to use that in the next step. Someone else, in fact, could also set up another pipette for 3 microliters. So you should be thinking steps ahead and setting pipettes to the appropriate amounts when you're just having downtime. Another thing is, we talked about removing the supernatant at some steps, and we wanted to keep the pellet. For example, if you look at this step here, this is where we had the lysis solution, and we want to get rid of the lysis solution, and we want to eventually leave the pellet and eventually add the wash buffer. But we have to make sure we get rid of all this supernatant above the pellet. So what you have to do is you have to think about how much liquid, approximately how much liquid is here, so that you're only left with the pellet. Well, if you go back a few steps, you can backtrack that. Well, there's 150 microliters that you added here. There's 3 microliters of the silica. So you're saying roughly, when I'm taking out liquid, I'm roughly taking out 150 microliters of liquid. So you can set your pipette for 150 microliters and just make sure not to disturb the pellet. I believe that happens again over here, but this time notice you have 500 microliters. In this step, you put 500 microliters. So when you're removing the supernatant, you want to set your pipette to near 500 microliters or 495 microliters. So you should think about these steps as you're proceeding.